Topic 1, Genes and Heredity, Lesson 3, Genetic Coding and Protein Synthesis, pages 26 to 35 in your textbook. All right, as always, we're going to start with the connected, this time on page 26. So a blueprint is a plan to build something. Circle the blueprint in the image. So yours should be circled here. Then we need to answer these questions. Make connections. When have you used instructions to build something? So be that using instructions to build something in Minecraft, using instructions to build, in quotations, a recipe, to build a desk. Anything that you've done, you should write your answer. I have used instructions to whatever it is you said. Then SEP designing solutions. How did the instructions help you with the building structure? So the instructions help me to complete the design. The instructions help me to understand each step of the process. Whatever you felt the instructions helped, and you should answer that accordingly. All right, so the genetic code, DNA or deoxyribonucleic acid, is the genetic material that carries information about an organism and is passed from parent to offspring. So of course, DNA is one of our vocab words. Now, sequence is the order that forms the instructions for building proteins. It's long chains of amino acids. And sequence is one of our academic vocabulary words. So in 1953, DNA was discovered. This looked like a twisting ladder called a double helix. Academic vocabulary, you need to list some other contexts in which you have seen the word sequence. So please do so in your workbooks now. DNA. So DNA is made of sugars, phosphates, and a nitrogen base. The sugar is called deoxyribose, hence where DNA gets its name from. There are also 4 nitrogen bases. Adenine, marked as A usually when we do any kind of DNA pairing. Thymine, which is the T, guanine, G, and cytosine, C. So now I want you to look at this image to the right. How does DNA pair or stay together? What pairs with what? So I want you to look at the nitrogen bases and figure out which ones go together based on color coding. When you're looking at that, you should notice that adenine always goes together with thymine, and cytosine always goes with guanine. And then there's the phosphate backbone, which is the side of the ladder. DNA replication. Scientists estimate that humans are made of approximately 37 trillion cells. The body needs to continuously make copies of DNA for you to grow. Why does the information need to be copied exactly? What might happen if there was a mistake in the DNA replication process? So DNA will need to be copied exactly so that way everything is the same and everything functions correctly. So our DNA codes for proteins. Our proteins tell us different things that we need to do. When there are mistakes in there, those will cause mutations, which is something we'll talk about later on in this topic. So continuing on, DNA replication. So to replicate or copy the DNA, it needs to be taken apart. So the DNA double helix will begin to untwist, and then a protein will unzip the DNA at the weakest part, which is the middle, where those nitrogen bonds are. So next, the nitrogen bases begin to attach one half of the DNA. They fit like a puzzle, so only certain ones can match with others. So that's what I was talking about, adenine with thymine, cytosine with guanine. And I just want to clarify, when I said nitrogen bonds, I'm talking about the nitrogen bases bonding together. This will continue till a chromosome with 2 identical DNA strands is formed. So they have to unzip, and then they recopy and replicate, and it's a whole fun process that we are going to get to learn today. So now it's time to do a reading check. So how is the separation of DNA like a zipper? And when you unzip a zipper, the two halves part. In the same way, when DNA separates, it breaks along the center into two halves, just like we see in this image here. So we can put it together, we can take it apart. All right, also, we have the design it. So you need to sketch how you would model DNA replication using household materials such as beads and pipe cleaners. How do the pipe cleaners and beads relate to the structure and function of DNA? So remember that you're using something as your nitrogen bases, or your adenine, thymine, cytosine, and guanine, and then you have something that's your backbone phosphate backbone for that, which would probably be your pipe cleaner. So make sure that you draw an image and label everything in that, please. All right, making proteins. So proteins are made from building blocks called amino acids. There are only 20 amino acids in the human body, but they code for thousands of different ways to make proteins. What is similar to this, and what uses around 20 items to make thousands? So what's similar to this is the alphabet. So we have our letters of the alphabet, and then they are used in different combinations to form thousands of different words. Protein synthesis vocab, the process by which amino acids link together to form proteins. All right, now this image is figure 4, so structure of DNA and RNA. There are differences between DNA and RNA, and they are apparent when comparing their structure. We should use the diagram to identify two differences between a DNA molecule on the left and an RNA molecule on the right. So RNA has the nitrogen base uracil, while DNA has thymine. RNA is a single strand, while DNA is a double strand. Now RNA. All information to code for a protein is located inside the DNA, which is inside the nucleus. But the protein building process takes place inside the ribosome. RNA is very similar to DNA, however, it can travel between the nucleus. RNA will work as many things, but in this instance, it will work as a messenger. So mRNA, messenger RNA. And its sole job is to copy the DNA and then take that replicated copy and take it to the ribosome to make more copies of it, because DNA cannot leave the nucleus. It's stuck. But RNA can. There are different types of RNA. So we just talked about messenger RNA or mRNA, and it carries copies of instructions for the assembly of amino acids into proteins from DNA to ribosomes in the cytoplasm. Note that a codon is every 3 bases of mRNA. Now, transfer RNA, or tRNA, carries amino acids to the ribosome during protein synthesis. An anticodon is every 3 bases on the tRNA. So here we have a tRNA molecule, or for figure 5 in our textbooks. We need to fill in the label to identify what tRNA molecules carry. And they carry amino acids. Protein synthesis. Transcription. DNA unzips to make mRNA. mRNA leaves the nucleus and travels to the ribosome. Translation at the ribosome attaches to mRNA and starts reading the codons. Every 3 bases code for 1 amino acid. tRNA brings the amino acids, creating a protein chain. A protein chain. Comparing structure of DNA and RNA. So again, DNA has sugar deoxyribose. It's double-stranded. It contains thymine, and it stays inside the nucleus. RNA has the sugar ribose. It's single-stranded. It contains uracil, and it can leave the nucleus. mRNA codon table, page 31. Knowing the code, figure 6. So a codon is a sequence of 3 bases that codes for one amino acid. For the DNA sequence CTA, the complementary mRNA codon would be GAU, because guanine pairs with cytosine, adenine pairs with thymine, and again, adenine pairs with thymine, but RNA doesn't have thymine, it instead has uracil. So scientists use mRNA codon table, the mRNA codon table, to determine which codons will code for each amino acid. The highlighted parts of the table show how the codon GAU codes for aspartic acid, also known as aspartate. So we look at the left-hand side for the first position for G. Then we find in the top row where our second letter A is. We look down where those two cover or meet up. And then we look to the right in the third position to find that U. And then where it meets up and all three kind of pass together, we see that we have aspartic acid. So what is the mRNA sequence for the DNA sequence ACC? We have UGG. And what amino acid is that mRNA sequence coding for? So we look for U on the left hand, then we look at G at the top, and then G on the third. To the right is tryptophan. Why would it be incorrect to say that the DNA sequence ACG codes for the amino acid threonine? So, if we see DNA ACG, we first have to do the RNA. So, ACG is the DNA sequence, not the mRNA sequence. You must first find the complementary mRNA sequence, UGC, then use the codon table to determine the amino acid cysteine. So here we have protein synthesis. Figure 7, protein synthesis begins in the nucleus and ends at the ribosomes. So at 1, we have formation of messenger RNA. Inside the nucleus, DNA unwraps and mRNA is made from the gene. Messenger RNA then leaves the nucleus and enters the cytoplasm. 2, ribosomes attach to mRNA. After arriving in the cytoplasm, a ribosome attaches to mRNA. The order of the base pairs on mRNA determines which tRNA molecule attaches to the strand. Each codon on the mRNA strand attaches to a complementary anticodon on the tRNA strand. Messenger RNA provides the code for protein construction. So just from going to 1 to 2, we remember that messenger RNA provides that code. 3, tRNA brings the correct amino acid. So the order of base pairs on tRNA determines the type of amino acid it carries. As the ribosome moves along the tRNA strand, molecules of tRNA bring their attached amino acids. So 4, a protein chain is formed. So as tRNA anticodon line up with mRNA coders, the amino acids bond on the ribosome and form a long protein chain. So protein synthesis, page 33. So our model it. Use the steps in figure 7, which we just went over, as a guide to fill in the missing molecules that drive each step of the process. Then complete the flowchart with the complementary nitrogen bases or amino acids. You should refer to the codon table in figure 6. So the first thing we're going to do is we see the TGTGAA. We're going to do this complementary RNA strand, and that'll be, well, first we need to label that that's DNA. Sorry. So we know that's DNA. So now we're going to do the complementary RNA strand. So messenger RNA, and then after the messenger RNA, we're going to have tRNA, and then we're going to get a protein. So that's our steps, right? It goes from DNA to mRNA to tRNA and then to protein. Now let's get into what things code for. So TGTGAA, it codes for ACACUU. Then the tRNA will code for UGU GAA, and UGU will code for the protein cysteine, and GAA will code for the protein glutamic acid. All right, now it's time for our lesson check. So CCC, identify patterns. List the 6 nitrogen bases that would pair with the following sequence of bases in a strand of DNA. So if we have TCGACA, we are going to get AGCUGU. Explain phenomena. Why does the cell complete DNA replication? So DNA replication ensures that each cell contains the correct amount of DNA to carry out life processes. Synthesize information. What will happen to a protein when the DNA codes for a different amino acid? And that would be that a different protein will be constructed. Support your explanation. Explain the importance of the sequence of nitrogen bases on a gene to heredity. So the sequence of nitrogen bases can be inherited. It directs the construction of proteins, which the cell uses to express the trait or perform a function. CCC, analyze structure and function. DNA replication begins when the double helix untwists and breaks in half between the nitrogen bases. What are the next 2 steps in the process of DNA replication? So the next step involves nitrogen bases with sugars and phosphates pairing up with the exposed bases on each half of the DNA strand. Then, with 2 new complete strands formed, each double helix twists into shape. 6. Explain the relationship of making proteins to inheritance of traits. So genes that are received from parents determine the proteins that are made, and in turn, the traits that are expressed. Finally, 7. Develop models. Sketch and label models of DNA and RNA that show how the difference in their shapes, their sugars, and nitrogen bases. So here we have RNA or DNA. So for the RNA, for your sketch, it does not have to be twisted. You could draw a straight line. That would be acceptable for this image. For DNA, it does need to have that double helix. If you need a better example of how to draw this, come ask me to see my book, and I'll show you how I drew it in my textbook. But this is a good example and good visual representation. So then we have to make sure that we include everything. So we need to label the RNA as single-stranded because that's one of the main differences, while DNA is double-stranded. Then we need to mark that it is made of ribose, while DNA is made of deoxyribose. Also, we need to differentiate that RNA uses uracil, while DNA uses thymine. And once we have that, that covers all of our bases, and that completes this lesson.